GR IAS Academy.
Hello everyone, welcome to GR IAS Academy. Today we are on the day 4 of Mission Surya Programme. It is a 100-day prelims marathon program in which we will be discussing 100 important current affair themes for prelims 2026. Let's go on to the first topic for today. So today's first topic is sickle cell disease. It is a hereditary blood disorder . It is a hereditary blood disorder caused by the mutation on the beta globin gene. So because of the mutation on the beta globin gene, this disease occurs. Mutation refers to sudden change in the DNA sequence in the gene that might occur due to environmental factors like exposure to radiation or might be during the DNA replication process. So whenever this mutation happens on the gene, the red blood cell, which is generally in disc-like shape, gets turned into sickle shape. This is a sickle shape or the crescent shape. So red blood cell turns into this shape. So what happens when red blood cell becomes sickle in shape? So generally when the blood flows in the blood vessels, because of the sickle shape, the blood gets obstructed in the blood vessel, so that oxygen carrying capacity of the blood reduces, less oxygen is carried to the organs. Because of that, organ damage occurs. And also, because of the sickle shape, the lifespan of the red blood cell reduces. A normal healthy red blood cell lives up to 120 days, whereas sickle cell has lifespan of only 20 days. And due to the reduction in number of red blood cells, severe anemia occurs. So basically, in the sickle cell disorder, oxygen supply to the organs reduces, severe anemia occurs. So let's see the prevalence of this disease in India. So if we see the prevalence in India, it is generally seen in tribal belt, that is in this belt. This region, there is high prevalence of sickle cell disorder. So what are the reasons why this disease is highly prevalent in the tribal areas? Because there are two major reasons basically. First one being endogamy. Tribals generally practice high rates of endogamy, that is they marry within their groups so that the gene gets circulated among them and finally gets expressed. So that is the reason why this disease is seen highly among tribals. Let's understand the genetic inheritance of this disease. This disease is a hereditary one, so it follows a recessive inheritance pattern. Let me explain this recessive pattern little clearly. So here there is a mating occurring between a male and female, and they are producing 4 offsprings. So whenever a child inherits genes are inherited in pairs. That is, one copy of the gene they get from the father, and one copy they get from the mother. So in this case, suppose this A represents the healthy gene, whereas this S is the mutated gene. Similarly here, the mother also has one healthy gene and one mutated gene. So whenever they produce offsprings, there are different probabilities of offsprings inheriting these genes. For example, here in this case, one person is getting healthy gene and one person is getting 2 diseased genes. And here if we see these 2 people, they are getting one healthy one and one diseased one. In this case, the inheritance is recessive. That means the gene gets expressed, that is the symptoms of sickle cell disorder is seen only when 2 copies of the gene are diseased. So the recessive disorder means only in this case the individual is deceased. this offspring has got 2 healthy genes. So here this is healthy child, whereas these 2 people who has got 1 healthy gene and 1 diseased gene, they act as carriers. That means they do not have any symptoms of the disorder, but they just act as carriers, that is they carry the affected gene within them. So this is meant by recessive inheritance. So another reason for tribals, another reason for high prevalence of this disease among tribals is that this gene provides a survival advantage for tribals. Let me explain this clearly. Tribals are historically forest dwellers. In forests, there is high occurrence of malaria. So when the treatment was not proper, tribals used to die because of malaria. So but in case of tribals, if they had this gene as 1 copy, sickle cell gene as 1 copy, they had shown immunity against malaria. That means these carriers, they have shown survival advantage against malaria. So natural selection has favored these carriers in the malaria prone conditions. So these healthy individuals might have died because of malaria. These diseased ones would have died because of sickle cell disorders. But these carriers were survived in the malarial conditions. So because of this, the population of these carriers has increased. So today in the tribal areas, many people are actually carrying the gene without expressing the symptoms. So that is meant by survival advantage provided by the sickle cell gene to the tribals. So because of the more number of carriers present among the tribals, that gene gets circulated when they mate and finally that gets expressed. So let us see So in order to tackle this sickle cell disorder, government has come up with National Sickle Cell Anemia Elimination Mission. It was launched in 2023 and it aims to completely eliminate the disease by 2047. There are 3 steps involved in this mission. Basically they are, first step includes awareness and premarital counseling. So we have seen there are many number of carriers for the disease in the tribal people. So we basically do the screening of the people, and when we find carriers, the genetic counselors counsel them and say that say the probability of having the offspring with this disorder if they marry each other and mate. So this is basically the prevention step in which we are creating the awareness and also doing the premarital counseling, and next screening and early detection. So here under this mission, government has aimed to screen 7 crore individuals for the presence of the mutated gene by 2026. So recently screening of 6 crore individuals have done, and in that many carriers and many diseased individuals were identified. And the third step is to provide care and support for the identified individuals. So this is all about National Sickle Cell Anemia Mission. Let's move on to the next topic for today. So today's second topic is sea foam. So let's understand. Sea foam, that is nothing but a froth or foam that occurs on the sea like this. It occurs when the organic matter dissolved in the sea water gets agitated by the action of wind or waves, and during that agitation process, the organic matter traps some air and forms bubbles or foam. That is seen as foam on the seawater. Generally this sea foam is not harmful, but whenever the toxic materials are entered into the sea through the organic matter that the foam might become toxic. For example, let's see the sources of this organic matter first. So where does the organic matter come into the sea? First one, maybe due to the decay of the marine organisms. And second one being the terrestrial discharge. So when the marine organisms decay, the dead matter or the organic matter gets dissolved in the sea. Or the second source being the terrestrial discharge. Terrestrial discharge can be industrial waste or the domestic sewage waste, or it can be agricultural runoffs. So these are the sources of organic matter in the sea, and because of this organic matter, finally sea foam is produced. It is generally harmless, but whenever this sea foam is present in excessive quantities, that is an indication of marine pollution. How? Because this organic matter can lead to algal blooms. Let's see what is algal bloom in the next slide. But whenever there is high quantity of nutrients and organic matter, this can lead to algal blooms. That is why presence of excessive sea foam can be an indicator of marine pollution. So let's see what is algal bloom. So the next topic is algal bloom. Algal bloom is nothing but the blooming of algae in the water. That is rapid increase of the algae in water bodies due to nutrient enrichment or the organic matter. So the sources of organic matter remain same again. The domestic discharge or the industrial waste or the agricultural runoffs. All these carry the organic water into the sea, and there, because of the presence of these nutrients, the algae percentage increases rapidly. Let's see the conditions that favor the algal bloom. For example, when there are warm temperatures and if the water is not moving or if the water is stagnant, and if there is presence of excess nutrients, these conditions finally lead to algal bloom. What happens if there is rapid increase in algae in the sea? These algal blooms becomes harmful. That is harmful algal blooms. They become dense, they get stressed and finally releases toxins. And these toxins are harmful for the marine life. And also, when these algae grow and finally decay, the bacteria decomposing this algae uses up all the oxygen present inside the sea and creates a hypoxic condition. Hypoxic condition refers to the condition where there is less oxygen available for the marine life, and because of that all the marine life gets killed. So it leads to fish kills and damages the ecosystem. And the rapid increase of algal bloom due to the excess of nutrients and organic matter is also referred to as eutrophication. So eutrophication is nothing but another name for algal bloom. I hope this is clear. So this is the next topic, that is CAR T cell therapy. CAR T cell therapy, that is chimeric antigen receptor T cell therapy, is an advanced immunotherapy in which T cells are genetically modified to attack the cancer cells. So in this therapy, we take the T cells and they are genetically modified in order to attack the cancer cells. So this therapy is used for the cancer treatment, especially in cases of blood cancer like leukemia and lymphoma. So let's see this therapy in detail. So before that, let's see what are T cells and what are T cells. White blood cells, we know that white blood cells provide defense mechanism in our body. Lymphocytes are the type of white blood cells which are responsible for the immune response. So there are two main types of lymphocytes, that are B cells and T cells. So B cells and T cells. Both these cells are produced in the bone marrow. So both of these cells are produced in bone marrow, whereas B cells gets matured in the bone marrow itself. That is why the name B, whereas T cells gets matured in the thymus gland. That is why the name T. So remember, T cells gets matured in thymus gland, whereas B cells gets matured in bone marrow itself. These B cells have antibodies on them and they directly attack the pathogens. That is, they directly attack the bacteria or virus present inside the body. Whereas T cells, they operate through the release of cytokines. So these cytokines are nothing but some kind of protein messengers. They activate the immune response within the body. So these T cells directly attack the problematic cell. For example, cancer cell. So B cells attack the pathogens, whereas T cells attack the cell, infected cell directly. Here we can briefly see the functioning of T cell. So this is a T cell. And for example, this is an affected cell or a cancer cell. So on majority of the cells, there are antigens. Antigens are nothing but some kinds of protein that is present on the surface of the cell. Whereas T cells have some kind of receptor. These are protein receptors. They just go and latch onto the antigens present on the cell. So here, when they get latched onto the antigens present here, they release the toxins. And because of the toxin release, this cell gets killed. So there are receptors, antigen receptors present on the T cell, and there are antigens present on the cells generally. So this is the action by which T cell generally kills the affected cell. So let's come to the CAR T cell therapy and the cancer treatment. So at present, for the cancer treatment, we generally use chemotherapy or the radiation therapy. But the problem with these therapies is that they not only attack the affected cells, they also attack the healthy cells. So we cannot precisely target the affected cells using these therapies. Whereas CAR T cell therapy is used to directly target the affected cells. So CAR T cell therapy, where CAR T refers to chimeric antigen receptor. So if we see, T cells already have some kind of protein receptors or the antigen receptors. They are also called as antigen receptors because they are going and attacking the antigens. In the CAR T cell therapy, we genetically modify this T cell and insert some special kind of receptors here, maybe like this. So we insert, already there are some receptors present, we insert some special receptors. These receptors are made from different biological components, and these special receptors are called as chimeric antigen receptors. So we take the T cells and we insert the chimeric antigen receptors to better target the cancer cells, and finally we release this T cell into the body. Let's see this process little in detail. So this is the CAR T cell therapy. Here First, we take out the T cells from the affected individual and we reprogram those T cells and introduce chimeric antigen receptors into the T cells. So here they become CAR T cells, and then we multiply those T cells in the lab and produce millions of such T cells, and those T cells are again inserted into the affected individual so that they go and better target the cancer cells. I hope this is clear. The T cells are reprogrammed and again inserted into the individual. So here if we see, we just attached some better receptors onto the T cell, that is chimeric antigen receptor. So if you see here, modified T cells are designed to recognize specific antigens on the cancer cells, and they are used in the blood cancer like leukemia and lymphoma. So the specific antigens now directly attack the cancer cells, and these T cells are kind of personalized medicine. Here we are not developing some generic medicine and giving to all the affected individuals. Here we are developing medicine according to that individual. So we are taking his own or her own T cells. So that is why this therapy is expensive, and we have seen that T cells during their functioning, they release cytokines. Cytokines are nothing but some kind of protein messengers. They activate the immune response within the body. So after this T cell therapy, high amounts of cytokines are released, which leads to cytokine release syndrome. So it is a side effect of the T cell therapy, but it is managed by the doctors using some generic medicine. And this is all about the CAR T cell therapy. I hope this is clear. So if we see the current status of this therapy in India, it is basically in the clinical trial stage. But there is an indigenously developed CAR T cell therapy, that is NexCAR19. This is approved by the Central Drug Organization and it is being used for the blood cancer purposes. So this is all about the CAR T cell therapy. Let's move on to the next topic. Next topic for today is mitochondria. We all know that mitochondria is a cell organelle and it does certain important functions within the cell. So if we see the characteristics of mitochondria, it is basically a double membrane bound cell organelle. So it has two membranes to it and it is responsible for the ATP production through the cellular respiration. So we all know that mitochondria is called the powerhouse of the cell. Why is it called the powerhouse? Because it produces the ATP within the cell. This ATP is nothing but a chemical. For example, it is something like a battery that gives energy to the cell. So ATP is nothing but adenosine triphosphate. It provides energy for the cell functions, and this ATP, the energy unit, is produced by the mitochondria. It converts food and oxygen into the ATP. So this conversion of food and oxygen into ATP by the mitochondria is called as Krebs cycle. So basically, before the formation of ATP, there are multiple steps involved. Just before the ATP formation, oxygen gets released and an additional phosphate bond is attached to the adenosine diphosphate. That is how ATP, the triphosphate, is formed. And that process is oxidative phosphorylation. See, the Krebs cycle and the oxidative phosphorylation are the terms related to ATP production by mitochondria. So if we see another characteristics of mitochondria, they have their own circular DNA. We know that DNA is present inside the cell, inside the nucleus of the cell, whereas mitochondria is a separate cell organelle, but it does have its own DNA and ribosomes. So this presence of DNA in the mitochondria actually supports a theory called as endosymbiotic theory. This is nothing but, it is a biological theory in which it is said that mitochondria was initially a bacteria that is present outside the plant or animal cell, whereas during the formation of the cell, the bacterium has, so the bacterium during the formation of a cell has entered and started living inside the cell symbiotically. So that is why the name endosymbiosis. Living inside the cell symbiotically, that is helping each other. Mitochondria is providing energy to the cell, and the cell has provided some place of residence for the mitochondria, and it is living inside, that is why endo. So as a separate organism has entered the cell, that separate organism does have a DNA. That is why mitochondria has a DNA, and the presence of DNA in the mitochondria supports the endosymbiosis theory. So if we see here, they have their own circular DNA and ribosomes, and it supports the endosymbiotic theory. And another important point regarding mitochondria is that the DNA in the mitochondria gets inherited by the offspring. But here, the mitochondrial DNA inherited by offspring is only from the mother, whereas the mitochondrial DNA from the father gets damaged during the fertilization process. So so that means mitochondrial DNA is maternally inherited. And if we see another characteristics of mitochondria, they do have cristae. Cristae are nothing but some kinds of folds present inside the inner layer, which facilitates the ATP production, and they also have some enzymes for the production of ATP. And this is all about mitochondria. So this is the last topic for today, that is Bharat emission norms. So what are Bharat standards? Bharat stage emission norms or the Bharat standard norms are nothing but the vehicular emission standards. So it prescribes certain emission standards for the vehicles. For example, vehicular exhaust generally contains these substances: carbon monoxide, nitrogen oxides, particulate matter, and the hydrocarbons. So with the Bharat stage emission norms, we progressively restrict the emissions from the exhaust pipe. So let's see the different stages of the norms. For example, let's see the evolution of Bharat stage emission norms. First stage was introduced in the year 2000, and different stages were later introduced, and finally in 2020 we have directly framed the BS6 norms from the BS4 by skipping the BS5. So we had no BS5 norms, we have directly had BS6 from the 4. That is to comply with the European standards. So in order to comply with the global standards, that is the European standards, and also to speed up the reduction of air pollution, we have skipped the 5th stage and entered the 6th stage directly. And this 6th stage was implemented in 2 phases. And in 2026 and 27, there is a plan for the introduction of 7th stage emission norms. So, if we see here, we are progressively giving restrictions on these emissions from the vehicles. So, how are these emissions gets reduced? That is either by cleaner fuel technologies or by better battery designs or by advancement in the exhaust emissions treatment technologies. Exhaust treatment. So, by imposing these Bharat stage norms, we are indirectly prompting the industries, the vehicle manufacturing industries, to focus on these steps. So, because of this, we have had fuels with less sulfur content, and there are experiments with respect to exhaust treatments, like there are catalytic reduction experiments, or there are scrubbers introduced in the tailpipe, before the tailpipe emissions. So, these are the advancements with respect to the pollutant reduction. Right? So, by focusing on these technologies, we can reduce the pollutants coming from the vehicles. So, that is it. We have seen that we are following the Euro 6 standards, and there is a small cue here. Whenever there is something related to cleaner environment or environment friendly technology, Europe generally stands as the forerunner. So, Europe generally stands as the forerunner. This point can be used during the prelims if you get confused between 2 options, that Europe generally stands as a forerunner in cleaner technologies. So, this can be used as one of the elimination tricks, but you have to observe in which areas Europe is standing as the forerunner. We will see a few more such techniques in the subsequent lectures. That is it for today. Thank you. We have seen the topics in short and crisp manner. For detailed notes, please subscribe to GR IAS WhatsApp and Telegram channels. Links will be provided in the description. And also subscribe to the GR IAS Academy YouTube channel in order to get the regular updates regarding the Mission Surya program. Thank you.